Okay, yeah, thank you very much. Um, Today, what I'm going to be talking about is I just want to start out by saying this is a very large collaborative effort by our team. And so it's over a dozen labs across the world. And so when I use the term we here, I'm referring to any one of these people. And I've tried to highlight on the slides which labs contributed to different data that I will be presenting today. And I'd also just like to start out by acknowledging, our different funding sources from the DARPA and the NIH, and our support from the Gladstone and the QBI and QCRG efforts. And so I don't think I need to convince anyone that it's a very important and pressing research question to understand the biology of SARS-CoV-2 infection. But one thing I just want to highlight right here is the role that phosphorylation plays in viral replication. And so phosphorylation signaling plays an important role in every aspect of viral replication. So here's a few figures that I took from a review on flu infection. And what these figures really illustrate is that phosphorylation impacts attachment, entry, and release. For example, in the case of flu, by manipulating PI3K signaling, MAPK, PKC signaling to enable the virus to replicate efficiently. And it's not a coincidence that viruses use phosphorylation to achieve this, their replication. Unlike, transcriptional or translational regulation, phosphorylation is a very rapid mechanism and a reversible mechanism to quickly modulate the host. And this can be crucial for the virus to quickly modulate the host towards its own benefit, but also allows for the host proteome to rapidly combat the viral infection. So this is one reason that phosphorylation signaling is very important in viral biology. And so one of the most powerful ways to monitor phosphorylation signaling is through using proteomic approaches. And so that's what I'm going to be talking to you about today. And here we've performed global mapping of phosphorylation changes upon SARS-CoV-2 infection by proteomics. And then we take those, the differentially regulated phosphorylation sites that we find, use state-of-the-art bioinformatics approaches to translate those phosphorylation site changes into kinase activity regulation, and then use that information, since there are many drugs targeting kinases, to prioritize drugs that might have antiviral efficacy for SARS-CoV-2 infection. So that's the premise of the work that I'm going to be presenting to you today. And so the founding experiment for this project was we started with Vero E6 African green monkey cells, and these were selected because they're extremely permissive to SARS-CoV-2 infection. And so we either infect these cells and then harvest after an hour of incubation or across 24 hours of infection, or we perform a mock infection for comparison and harvest at the beginning and the final time point. And all of these time points were collected in biological triplicate. And then we analyze these samples in two different modes, one looking at protein abundance changes and the other one looking at phosphorylation changes. And finally, since we're using a monkey cell line here, we then map all of these phosphorylation changes to their orthologous human proteins so that we can use the wealth of, annotated resources to understand the changes that we're observing here. And so just to convince you that this data is of reasonable or high quality, one thing we can do here is from the protein abundance data is look at the infection. So we can detect many of the different viral proteins in our samples, and we observe a significant increase in the abundance of the viral proteins over infection. So we have a good infection here. And then also we have high quantitative reproducibility. So when we look at replicates of a sample within the same condition, we're observing Pearson correlations of usually 0.95 or better. And so what we observe here, when we look first at just protein abundance differences, is that over the course of infection, we see increasing amounts of proteins that are differentially regulated. And here I'm showing proteins that changed by twofold or more, and we're had a p-value, or excuse me, an FTR value of less than 0.05. And but we only observe about 160 proteins out of approximately 4,000 detected in this experiment that are significantly up or down regulated over the course of infection. And if you compare that, for example, to phosphorylation sites, here we observed 10 times the number of differentially regulated species here, with approximately 1,500 phosphorylation sites significantly regulated. So this is really demonstrating that phosphorylation represents a primary host response in SARS-CoV-2 infection. And one of the first things we did was look at the overlap in regulated phosphorylation sites and between this experiment and our recently published SARS-CoV-2 protein-protein interaction map. So for those of you not familiar with that work, there we expressed 26 of the SARS-CoV-2 proteins, and they were all strep tagged, and we expressed them in hex cells, and then we performed affinity purification to detect the interaction partners for those proteins. And then we look in our phosphorylation data set and say, okay, for NSP5, for example, we found this to be interacting with HDAC2 from our protein interaction map. And now we can look and say, do we see phosphorylation sites on HDAC2 that are differentially regulated? And indeed, for example, here, we see S394 of HDAC2 to be dramatically increased in phosphorylation almost immediately in the time course of our infection. And so the time course of infection here is shown around the circle here. And so we can do this for all of the baits that we analyzed in our protein interaction map. And what we find is that for 19 of these baits, their protein interaction partners are significantly regulated at the phosphorylation level. So this really suggests that these protein-protein interactions likely enable, help to drive changes in phosphorylation. The mechanisms of this change may be different based on those interactions. Some may be driven perhaps by changes in protein localization, but many of these interaction partners are being modulated at the level of phosphorylation. And so just going back to the overall design of the experiment, so another thing we really wanted to do from this data is to extract information about kinase regulation. And our collaborator, Pedro Beltrao, has developed a really nice tool called Phosphate, which contains millions of different mass spec experimental results on how phosphorylation sites change, and also annotated specific phosphorylation sites on substrates to their kinase. And what this tool enables you to do is input phosphorylation changes and export out predicted kinase activity changes. So we use this tool to predict or estimate the kinase activity changes upon SARS-CoV-2 infection. So here we have a heat map over the infection time course from 0 to 24 hours, and then a list of some of the most highly activated in red or inactivated kinases in this experiment. And so I just want to go through a couple examples here of the biology behind some of these differentially regulated kinases. And so one that we were really interested was we saw a dramatic and sustained increase in 2 subunits of CK2 kinase upon infection. And this is really interesting to us because from our protein interaction map, we had observed that N protein from SARS-CoV-2 interacts with 2 different subunits of CK2. And then from our phosphorylation mapping, we observed that several substrates downstream of CK2 have dramatic increase in phosphorylation, as shown on the right here. And many of these substrates are known to participate in cytoskeletal reorganization. So to investigate this biology of SARS-CoV-2 infection, we did some microscopy here, looking at F-actin, which is a component of the cytoskeletal system. And so here we find a couple interesting things. So one, when we infect cells with SARS-CoV-2, we observe the development of these philopodia here. And what we observe is that the presence and length of these philopodia is dramatically increased when we have SARS-CoV-2 infection. And within these philopodia, if we zoom in more on these immunofluorescence microscopy images, we observe the N protein in red co-localizes with CK2 protein in approximately 40% of instances. And so the virus seems to be, this protein-protein interaction can be happening at these new philopodia that are induced by SARS-CoV-2 infection. And then we collaborated with Elizabeth Fisher at the NIH, who does electron microscopy. And here she's done transmission electron microscopy, where we can really zoom in on these philopodia. And what you can see here is that this is a philopodia here, and we can actually observe a viral budding from this philopodia. So it seems that this interaction between the N protein in SARS-CoV-2 and CK2 promotes these philopodia that allow for viral budding and an easier mode of transmission of virus from cell to cell. So this is a really, ominous finding for the ability of SARS-CoV-2 to transmit between different cells. And finally, to just look a little bit more into the mechanism, we apply the same phosphoproteomics approach that I described earlier, but here instead of doing SARS-CoV-2 infection, here we just overexpress N protein alone. And we observe that N protein alone is sufficient to increase the activity of CK2, and also causes decreased activity of other proteins, such as some MAPK proteins and CDK1. And lastly, the goal of much of our work is to find drugs that could be used as treatments for COVID-19. And so we teamed up with Kayvon Shokat, an expert in kinase drugs, and he helped us map different drugs that may target CK2. And one drug, as shown here, is actually in phase 2 clinical trials. And so we performed some in vitro assays looking at cytotoxicity and antiviral efficacy here. And here we can see that this drug has 2.4 micromolar efficacy. So this demonstrates really that SARS-CoV-2 infection relies on N protein, modulation of CK2 for its survival. Additionally, we found downregulation of many cyclin-dependent kinases. So as shown here in the heat map, we have CDK2, CDK5, and CDK1, which are all downregulated in activity upon viral infection. And so to kind of probe this a little further, we assumed that this might change the distribution of the proteome and what stage of the cell cycle it's in. So we performed a cell cycle assay, and indeed what we find is that upon SARS-CoV-2 infection, which is the red distribution here, we see an increase in cells at either S or G2M phase. So this looks like SARS-CoV-2 infection is inhibiting cell cycle progression. And then again, to map this to drugs, we mapped this to several different drugs and tested, for example, one drug that's in phase 3 clinical trials here, and we find that it is very potent with submicromolar efficacy in these antiviral assays. And then one last biological insight that we observed was that there is an increased activity in the P38 and MAPK signaling pathway. So that it involves many of these MAPKs here, which by 24-hour time point are dramatically increased in abundance. And many of these, and the P38 signaling pathway is known to be involved in the inflammatory response upon viral infection. So this is not necessarily unexpected. And we further confirmed this increase, this predicted kinase activity increase by Western blot of phosphorylation sites known to regulate P38 kinase activity. And so the next thing we did was then find out which drugs we started we performed assays just here using siRNA knockdown of many of these P38 and MAPK signaling pathway components. And we find that in all cases, knockdown of different components of this pathway reduced viral infection. And then we tested several different drugs targeting different components of this pathway. And I just have one shown here, ralimitinib, which also has sub-micromolar efficacy in this assay. So in total, we didn't just do this for these few examples. Here we mapped dozens of drugs to the kinases that we find dysregulated under infection, and found many other cases of drugs that seem to be effective in inhibiting SARS-CoV-2 infection in these in vitro assays. So in total, we were able to map 87 drugs to dysregulated kinases and test 68 of these. And what I think is very pleasantly surprising is that for more than half of these drugs, we found that their cytotoxicity concentration was far greater than their IC50 concentration, making them potential candidates to move forward with testing. So in summary, what we did here was perform a phosphoproteomics assay over SARS-CoV-2 infection, and then to take that wealth of phosphoproteomics data and make it more translatable, we converted it into kinase activity assays, where we observe dramatic rewiring of cell signaling. We observe activation of CK2, which localizes at these viral-induced filopodial protrusions and seems to be a way for the virus to propagate from cell to cell. And then we take all this information and translate into drugs and demonstrate that many of these drugs have antiviral efficacy. So with that, I'd like to just thank again all of our collaborators, and I'd be happy to answer any questions.
All right, Danielle, thank you very much for that beautiful story. Lots of new targets, lots for people to work on. So the CK2 one is of interest to us. We think a lot about cytoskeletal changes induced by viruses because we know HIV does fiddle with the cytoskeleton also. So that's fun. Is it, and certainly the cell cycle stories are interesting. Is it curious that the virus is downregulating kinases by itself and that your inhibition of them makes the virus less happy? You would think it might make it more happy.
Yeah, I think that's an interesting one. And I think, you know, there's potential ways to think about that, for example, with the cell cycle kinases, that since these are cyclical in nature, if you just hit them and wait, it'll be effective. But it is interesting that in other cases, we do find that inhibition of things that are downregulated are effective. And it's not clear exactly why that works, but it seems to work.
Yeah, the CDKs are also important kinases outside the cell cycle, so they may be doing stuff unrelated, I suppose, to the cell cycle. But it's still interesting that the virus is knocking them down, and your pharmacological knocking them down is not helping the virus, it's actually inhibiting the virus. So that's interesting. All right. We have a couple of other questions from the audience. One person asks, what software pipeline did you use to look at the, I guess, changes in phosphorylation?
Yeah, for this we used the Spectronaut software, since we used a DIA acquisition approach, and then used the MS-Stats package for statistical testing.
Okay, those are programs beyond me. I guess the person can get in touch with you for details of how to do this.
Sure. And all this work has recently been published in Cell, so those details are in the method section of the publication.
Another person asked how the interaction of N protein with CK2 works. That is, how does it activate?
Yeah, I think this is really interesting, and I hope that someone, whether it's us or someone else, figures this out, because we've actually have now in-house data looking at infection in a panel of 3 more human cell lines here, And we find in every instance that CK2 is activated. So this seems to be a very conserved mechanism for SARS-CoV-2 infection.
Is it likely direct? I mean, can you add N protein to CK2 in vitro and activate it, or is it way more complicated than that?
That I don't know yet. And CK2 is a multimeric kinase. So you have there's usually, I think, 4 subunits.
Right, so it could be complicated.
Yeah, exactly.